Hello, everybody, and thank you for inviting me to the meeting. Too bad I can't be there in person. What I'm going to do today is talk to you about how we think phage lead to coral reef decline. So the question really starts at, when is it good for a virus like a phage to hang out in a cell? And really, I know everybody here knows all of the steps of different things like the lytic versus the lysogenic cycle. So what we're really focusing on is how much time does the virus choose to spend inside the host cell. And remember that we kind of artificially break this into two things where we would say viruses that go in and just blow up the cell as fast as they can are virulent, and ones that hang out in some sort of symbiosis are temperate. And then finally, just to remind everybody, when the virus is in the cell and hanging out as a temperate virus, we're going to call it a provirus or a prophage. And what's interesting about this behavior is once something has become a provirus, it's going to replicate at the same rate as the host cell. And that host cell plus virus is called a lysogen. And as far as we can tell, almost all cells are actually lysogens. Okay. The other interesting thing about this dynamic is that proviruses, whether they're phage or those hanging out in our cells, must protect the cellular host against other viruses and protists, or they'll just be killed off by those predators. So anytime that you have a system, an ecosystem with a relatively high abundance of viruses and cells, what you expect is to have a lot of anti-predator things built into these lysogens. These come in basically two flavors. So there's what we would call superinfection exclusion, which is something that would protect the host cell against other viruses. And then you have virulence factors, which are things that protect the cells against other protists. And then finally, when we're thinking in kind of the ecosystem version of this dynamic, we call this piggyback the winner, which basically says that as an ecosystem, as you get more and more cells in an ecosystem, you're going to get more temperate behavior. So they're going to hang out, the viruses are going to hang out with the bacteria or the eukaryotic cells for longer. Like, think in your body, there are so many cells there that if a virus starts replicating in a lytic sort of event, it's going to kill all of the cells, and that would kill any virus that happened to be a temperate one. Okay? And what we know now is that really the main switch between virulence and temperate behaviors is really controlled by the metabolic state of the cell. And the really easiest way to think of this is that it's just the amount of organic carbon available, so think sugar, divided by the amount of oxygen. Okay? Really, if you think of it in that terms, extra organic carbon, so extra sugar, will lead to more temperate viruses, whereas extra oxygen will lead to more virulent behavior. And we know this from like old studies of the Lambda switch. So this just shows you the two states. So in this case, if you have a cell where there's like a low carbon supply, there will be plenty of oxygen. So you can think of it that way. So it can be either that we don't have as much food or we have a lot more oxygen. If that happens, what the cell does is oxidative phosphorylation, and it produces a whole bunch of ATP. And there's a set of cascades, cellular signaling cascades, in this case, which encourage the phage to be a lytic sort of phage. If, however, you reduce the amount of oxygen or you increase the amount of organic carbon going into the system, the cell will effectively not produce as much ATP, And in fact, it produces all of the things that you use to make a new cell. So that's kind of the biosynthetic side. So this switch between these two things are basically catabolic. Think of that as extra oxygen, means lots of ATP, means lytic virulent behavior by the viruses. Anabolic, which would mean not enough oxygen or more food, produces a lot more NADPH, and that's what leads to the temperate behavior. And this just gives you an example of a lot of how we look at things. So we count a lot of viruses and microbes using direct microscopy. And really what you do is you just filter down a sample onto a 0.02 anodisk, stain it with cyber gold, and then count under an epifluorescent microscope. And when we do that, we find that all these little dots are the viruses. These bigger dots are your microbes. And we associate a low VMR, so that's the virus to microbe ratio, with the temperate lysogeny decision, versus a higher VMR is associated with the lytic virulent life cycle. And that's because when you're in the lytic phase, not only are you producing 25, 50 viruses per burst that happens, you're also killing off the cells. So that's this ratio actually artificially inflates that signal, which is nice. It makes it easier to see. And this just gives you a feel of what it looks like across a whole bunch of systems. These are just direct counts from a whole bunch of about 20 ecosystems representing thousands of different samples. And what we see is that once you start getting to these higher microbe numbers, the number of microbes per gram or milliliter of sample, you actually get a very low VMR. And when you're in kind of this mid-range of like about 10 to the 6 cells per mil, you see a lot higher VMRs, and that's because that's where you're getting some sort of lytic production. And an important thing to remember is that usually most microbial, viral ecology has been done in this sort of range, and many fewer studies out here. And so there's a little bit of a bias in the literature about how we think about it, or we're thinking more about lytic behavior, but actually the majority of stuff is happening at these higher densities. Okay. And remember that there's the induction event. So where the provirus decides to leave the host cell and then re-enters the lytic cycle. And just to show you how this works in the case of when we're just thinking of it in terms of metabolic state, here's an aquarium, and we're going to take the aquarium and monitor it over time. And then at some point, we add oxygen. We're going to filter down the cells and the viruses, count them using the direct counts. And what you can see is just by adding oxygen, we change the VMR from somewhere like 12 to close to 25. And that's just the induction event. So really we're causing the viruses to become lytic just by adding oxygen. We don't think this is because of direct DNA damage. We just think it's just because we're changing the redox state of the host cell itself. So how does this work on a coral reef? So if you're not familiar with coral reefs, the way that a coral reef is built is that you have the coral animal laying down calcium carbonate in the form of aragonite, and then coral encrustose algae are these cementing sort of algae. And these guys build giant structures, so that's how you get coral reefs that are literally thousands of kilometers long and several kilometers down into the depths. So they're really big. And on a healthy coral reef, these are the main things that are growing on the bottom. So these are things that have access to the sunlight. There are, of course, different types of algae. So that we're going to call these seaweeds just to make it easier in this case. And these are kind of macro fleshy algae as well as turf algae. And they're there, but they're the things that like the fish and the and the invertebrate grazers like to eat. So what happens is that they're always kept very small. And so sea urchins and grazing fish always keep the seaweeds in control, and then through a grazing event. And then these things are eaten by predator fish. So you get groupers and you get sharks. So what you have is you have basically a shunt directly from photosynthesis through the grazers to the big predators. What that means is that the microbes that are living on the reef, on the surface here, are actually controlled by the amount of food that they get from the coral animal and from the CCA. And of course, we know the coral's a little bit better. So this is effectively a mucus, just like you have in your gut. And that's being shed and fed to the microbes as dissolved organic carbon. But just, again, think of it as sugar, right? All right, so the first thing that humans do is we fish out the big things. And this just gives you an example of what this looks like. So in gray are places where it's harder for humans to get to fish. Where it's white is where humans are living and or it's easier to fish. And these are just different places in the Central Pacific. And then on this axis is the biomass. And what I want you to notice is that basically humans fish the big things first, and then they start fishing some of the smaller things. And you see a really dramatic decline in the amount of biomass represented by the fish on a coral reef that has been fished. And this effectively just opens up a whole bunch of energy that from photosynthesis that can feed the microbes. And this is what we call microbialization. So imagine once you stop fishing here, you reduce the predation on the grazing invertebrates and smaller fish. And so these guys are no longer being turned over as quickly. So you have stopped shunting energy up to the sharks. And instead, you see this expansion where the seaweeds get bigger and they start producing more and more dissolved organic carbon. This goes back and it starts feeding the microbes. So instead of the microbes being dependent on the coral, they're now getting their energy from these seaweeds. And this creates what we call a positive feedback system, the DAM model, which just stands for dissolved organic carbon, disease, algae, and microbes. And it's a positive feedback because as you start getting more microbes, they start killing corals, which creates more space for seaweed, which creates more food for the microbes. And as you get going down this direction, you cause the system to eventually switch from coral and crustose algae to these seaweed-dominated systems. We look at these all over the world. And the main types of data that we gather is we'll go out and we get water, and that's where we count the microbes and viruses, and we measure the amount of organic carbon and oxygen. And then we also do these metagenomes where we just we gather water from the benthos, and then we use that for viriomes and metagenomes. And then finally, of course, we get how much coral and algae is on the bottom, as well as fish counts. And we've done this for over 200 sites throughout the Pacific, parts of the Indian Ocean, primarily Sri Lanka and the Caribbean. And the general take-home message is that what you see is that glycolysis and that full production of ATP that's the sort of metabolisms that the microbes on a healthy coral reef are doing. And what that means is that all of that ATP is also being used to keep the phage populations more virulent. Okay? However, when we go to degraded coral reefs, we see this switch away from ATP-driven metabolisms to these NADPH sort of anabolic metabolisms, which is what we know causes more temperate behavior. And we think that it's actually pretty simple how it happens. It's just a physical difference. So in a coral, the way the coral gets its energy for is by having a single-celled algae called zooxanthellae that live in the coral animal. So what happens is that the algae inside the animal does photosynthesis, and it releases both the sugar and the oxygen, and they dissolve out into the coral animal. And so there's a very tight coupling between sugar and oxygen. So that's what we call the coral holobiont. So this symbiosis is really important. However, in the case of the algae, what we have is you don't, of course, have the animal. So in the case of the seaweeds, so what happens is that you do photosynthesis and the oxygen forms bubbles and it's bubbled away and it's exported out of the system. So in this case, we don't have enough oxygen, but we have lots of sugar. In this case, we have a strong coupling between the sugar and the oxygen. And that brings us to the other really important point of what the phage are doing. So remember that the proviruses or the prophage must protect their cells against invading things that would kill off the host cell when they're in the provirus stage. So the one that's probably most important here is the anti-protist sort of dynamic. And the way that that works is the prophage encodes things that are virulence factors, which will kill eukaryotic cells like the protists. Okay. Here's some examples, and probably everybody knows that essentially all bacterial pathogens are actually just lysogens that are carrying phage and or plasmids that have these exotoxins on them. These are just some of the ones, so the ones in red are things that we just observe a lot in the environment, whether it's a human associated or not. And remember that the exotoxins just kill eukaryotic cells. It really doesn't matter. They'll kill a coral cell just as well as they'll kill a protist or a human. So what we think is going on a fish reef is that because of this change in the amount of sugar versus the amount of oxygen, we are just developing more potential pathogens. And that's what's causing the coral reefs to decline. So because they're killing off the corals, these emergent pathogens. And this just gives you some examples. So what we find is that when we look at the number of microbes with the direct counts versus the number of viruses, we see that unlike you would predict from a kill the winner sort of dynamic, the number of viruses actually go down as we get the number of microbes, which is indicative of the temperate behavior. We also know that the number of species of microbes does not increase. In fact, that actually goes down, which suggests that you're not actually getting just more diversity. So to explain that change in the virus to microbe ratio. The other thing is that the protection systems against viruses, like restrictions in enzymes, CRISPRs, etc., also go down. So we don't actually see a strong evidence that the cells themselves are more resistant to the phage. Okay? But what we do see evidence of is an increase in genes associated with the temperate behavior. So what this is this is just going into in the virions from these samples. So remember, we've gone to these reefs, we've sampled out the viruses, we've isolated the viruses and sequenced them, and we just look to see, do we get more temperate viruses as the number of cells per mil increase? And that's what we observe. And we do that by both quantifying the amount of integrase or the X size, which are the two main proteins causing in the integration event. Okay. All right, and then finally, we also look for these virulence factors. So do we see more of those on the degraded reefs? And we do. So we think that what's going on is that the prophage are really creating these opportunistic infections, and they're doing that, or these opportunistic pathogens, which lead to coral disease, and they're doing this really to protect themselves against protists, not explicitly to eat corals. So we think that coral disease is an emergent property of how phage and their host cells are interacting. So the main take-home points are that overfishing on coral reefs leads to this decoupling between sugar and oxygen, which I call the Goldilocks point. And that leads to this dynamic where you get more proviruses hanging out in the microbes. Those guys, the proviruses are expressing virulence factors, which then cause disease, basically to protect the cells against protists. This is just a little bit more putting it together for people that are thinking about in their system. So really what our working hypothesis right now is that the lytic to temperate switching is one of the main drivers of community dynamics. And that what we think is happening is that as you start to get more cells, you get those cells eat up oxygen. And so you get more of this anabolic behavior. You see the lowering of the virus to microbe ratio. And that's where we get the piggyback the winner sort of dynamics. If you add back oxygen, as I showed you, you actually cause induction, which is why at these, like where you've got kind of closer to the equal amount of sugar versus the amount of oxygen, you get more lytic behavior, probably through induction. And what this does is this causes the ecosystems to switch very quickly when you change the amount of oxygen. There's another dynamic which we do not understand very well. It's called piggyback the loser, which is where the viruses are caught in cells that are at very low concentrations. Okay? And we don't really understand how that works, but we do know that if you have kind of the losers, the ones that are really there at low concentrations, if you add back a little bit of food, they will start to do DNA repair, which is sensed as DNA damage, and the virus is induced at this point. Okay? So we think these two dynamics are super important, though we don't understand piggyback the loser as well. And with that, I'd just like to mention, of course, my lab, which there's a ton of people from it. Cynthia was one of the main people involved in this work. The math people, of course, which helped me with all of the modeling and statistics and so forth. These are people in different areas that have helped us collect samples all over the world. Rob Edwards is the person that does all the bioinformatics. Mark Hatte builds all of the things that we use for sampling, and Anka Segal is my the phage expert. Okay? And then finally, these are the people that would pay for it, and I'll be happy to answer any questions from anybody.